Hello guys, and welcome to another episode of biology. And of course, today we're looking at Mendel's work. And of course, we're going to basically look at monohybrid cross only, all right? In the next episode, we'll talk about dihybrid cross, all right? So let us just say we're looking at Mendel's laws, okay, of genetics. And then we're going to look at monohybrid cross only in this class, okay, so that we do not spend too much time. All right, so please do not forget that these tutorials, all of this episode, are brought to you by the one and only O3 Schools app. Okay, if you have made a conscious decision to pass your exam at once, either JAMB, WAEC, NECO, GC, NAPTE, any O-level exam together with JAMB, all right, these videos are for you, all right? So do not think it's only for JAMB. Despite we usually just do JAMB, okay, does not mean that the lessons are comprehensive. All right, you will not see this anywhere, trust me. Okay, so please ensure you want to jump on this opportunity, this sheer opportunity to learn very well and succeed. Okay, so please, we're going to see a question from the O3 Schools application. It's a JAMB question. All right, so please, this is a 2020 question. Yeah, 2020, and then number 11. Okay, so it is being displayed on your screen right now. It says here that which of the following represent the phenotypic ratio when a plant RR, right, is crossed with another plant RR, assuming that the gene R for round seed is dominant and wrinkled R is recessive. Okay, so which of the following represent the phenotypic ratio when plant RR is crossed with another plant RR, assuming that the gene R for round seed is dominant and the wrinkled R is what? Recessive. A says 1, 2, 1, B says 2, 2, 1, C says 3, 2, D says 3, 1. All right. So we'll see this question and many more at the end of the class and not before, because at O3 Schools, we pride ourselves, okay, in giving you value first. We believe that once we have taught you properly, then to answer questions will be the list of your problems. All right, so please ensure you stay close to us, all right, and download the O3 Schools application now. All right, you do not have any choice. Okay, download the app. I don't care if you have other apps on your phone. All right, please, this app is a solution. Okay, O3 Schools is the way to school. Okay, if you want to enter into school, O3 School is the way. All right, it is the sure path for you. Download the app, follow our tutorials. There are tutorials on the application also. Okay, download the app and you see what all of the benefit. The app has everything you need to succeed in your exam. All the past questions for all the subjects are on the application. What is more, you have wonderful features like the classroom feature. The classroom feature has the latest JAMB syllabus, has lecture notes, has video lessons. There's a study plan on the application also. Okay, what the study plan does basically is to create a timetable for you using the syllabus, all right, and the note on the app. That means a lot of you made the mistake of preparing for your JAMB without finishing your syllabus, and that's a big problem, right? It's a big problem for you. If you do not finish your syllabus, you are most likely to fail. All right, so please ensure you finish your syllabus equitably. Okay, if you want to perform excellently well, finish your syllabus. Okay, so to create a timetable for you within the time frame that you have chosen to finish out your syllabus. If you say in the next 1 month I want to finish my syllabus for biology, okay, or I want to finish my syllabus for my 4 subjects in the next 1 month, or in the next 2 months, the next 3 months, 4 months, 5 months, depending on what your target is, it will use the time you have given the app to plan for you, create a timetable for you. Every day what you read will be shown to you. If you're able to follow it, you finish your syllabus in time. Okay, wonderful feature. Then there's a question search feature where you can search for questions, just like the way we search for this question right now. Okay, went to the app, click on question search, click on biology as a subject, then search for what? Mendel's law, or search for monohybrid cross. Anything you want to search for it. All right, question that JAMB asks that related to any of this keyword that you have searched for will come out for you to answer. Also, there's a mock challenge on the application. All right, so every Saturday, students come to the application, either the JAMB or the WAEC application, to compete with each other to win amazing prizes. But the reason why we do that challenge is not because of the prizes, because many of you are usually distracted. The reason is not because of the prize. We are just trying to create a competitive edge for you, because a lot of people will know that their spark, okay, they see their spark when they are competing with their mates. All right, so when you compete with your mates, You will get used to JAMB past questions or WAEC past questions on the WAEC app, all right? You get used to likely exam questions. You familiarize yourself with how JAMB or WAEC set question to put you in place of the examiner. Only you will be, once you read, you only be saying, this question will be like question of the set, okay? You'll be able to discern what questions by yourself. All of these are on the application, all right? And then there's an activation fee that is less than the price of rice and chicken in a good restaurant, all right? If you want to succeed, all right, and not succeed, please ensure you download the application now. Activate it, okay? Let's say this phone number on WhatsApp. If you want to get your activation key, okay, then if you want to activate it by yourself, just click on activate on the app. The next page that will open, you'll see buy activation key. Click on it. You see the options for activating. Make your payment, okay? Send the receipt to this number on WhatsApp. You'll get attention. All right, so because of time, let us just continue with what we have for today. So Mendel's laws and monohybrid cross. Okay, basically, we're going to be looking at all the work that Mendel did. Okay, and we're going to see all of the scientists, okay, that worked, okay, on genetics, okay, and what on variation and all the likes. All right, and so that we will not be because sometimes JAMB will ask a question, or I could ask a question, which of the following scientists is not related with genetics. Okay, so you can be able to discern. Right, we do not want to leave any stone unturned. Right, so now we have looked at many definitions. We looked at alleles, we have looked at heredity, we have looked at genes, looked at DNA, we have looked at heterozygous alleles and homozygous alleles, dominant alleles and recessive alleles. We have looked at all of those things. And the reason why we're able to do that is because of the foundation work that Gregor Mendel did. Gregor Mendel, okay? So it's because of the work that this man did from 1822 to 1884, okay? Because of the work that this man did that we're able to talk about all of these, all right? And then, of course, Gregor Mendel was from a very poor home. He was born in a very poor home, okay? He was born to poor farmers, okay? In Austria, okay? Austria. All right, so his parents were poor, so he grew up working in the farm with his parents. But Mendel was a very curious person. He was very curious, eh? And despite the fact that they were very poor, all right, his parents from an early age they saw that this boy he know what they do. And so, despite their very poor financial life and despite their financial struggles, they saw that this boy, okay, is very intelligent, all right? So they decided, okay, they struggled to send him to school. Mendel did well in primary school, okay? He did well in primary schools, and then he was sent to grammar school, okay, in Tropau, all right? So, of course, he was a very bright student, but of course, he suffered a lot of depression, okay, because of financial struggles, all right? Because of the financial struggles, all right? Mendel faced a lot of depression, but later he joined the Philosophical Institute, in Olomouc, all right, studying physics and mathematics, okay, studying physics and mathematics, okay, and of course, despite all of these financial struggles, all right, he kept on keeping on because of because of his curiosity and his knack, all right, for knowledge. But at the age of 21, okay, he joined the Augustinian monastery, okay, all right, at age 21, all right, he joined the Augustinian monastery. And why did he join the monastery? All right, he was poor, okay, so the monastery provided free education, okay, even free food, all right, and the monastery had a garden, all right, they had a lab where he could perform his experiments. And there was even a library, okay, all right, in what? In the monastery, all right? And it was even in this monastery that he was given the name Gregor. Gregor, okay? So his birth name is not Gregor, all right? So it was a nickname that was given to him, or a sobriquet that was given to him in what? In the monastery, all right? His birth name is Johann. Johann, all right? So Gregor, it was a name that was given to him in the what? In the monastery. From the years 1851 to what? 1853, when what the monastery saw his talent, okay, they sent him to the University of Vienna, all right? He was sent to the University of Vienna. University of what? Of Vienna, all right? So when they saw his talent, okay, they saw how good he was, all right? They saw his curiosity, they saw his drive, right? The monastery sponsored him, okay, to the University of Vienna to study physics, okay, mathematics, and what? And biology. Okay, so but at the university, he majored on what? On statistics. Okay, in mathematics, all right, he learned about statistics, he learned about what? Probability also. Okay, which made him to be able to bring that knowledge to perform his experiment, okay, at the monastery. And in the years 1856 to 1863, okay, Mendel now worked on what? On the pea plant, PEA plants. Okay, he conducted his experiments using what? The pea plant, okay? And the reason why I used the pea plant was obviously, just like we saw for the Drosophila melanogaster, all right? Some of the reasons were also some of the reasons why I worked on what? On the pea plant, all right? I'm going to see all of those reasons very soon, but of course, they're easy to grow and they were to produce a lot of offspring, all right? And they do not have a very, very long lifespan, all right? They have a short lifespan. They grow what? In time, in a very short time, all right? So some of those reasons were the reason why he worked on the pea plants. Okay, so Gregor Mendel worked on the pea plants, all right, using all the knowledge he has gained from his journey in the University of Vienna, where he studied physics, mathematics, and biology, all right, and learned about statistics as well as probability. All of this work led to to the laws that he was able to bring out, all right, to a very big discovery. All right, we are going to look at very soon, all right, in this class, all right. So it was one that introduced the law of segregation, all right, then the law of independent assortment. All right, so Mendel published his work in the year 1866, okay, but it was not given much attention at that time. All right, when he published his work in the year 1866, his work was not given much attention. All right, but some years later, his work also was rediscovered, all right, especially in the year 1900, all right, his work was rediscovered. And of course, Mendel died in the year 1884, all right, without knowing that his work, okay, all of the work he has done will lead to to a very solid foundation, okay, in the world of genetics, okay. So his work was rediscovered in the year 1900, and today, all right, today you and I know Johann Gregor Mendel, all right, as the father of genetics. Okay, so first of all, what is genetics? All right, what is genetics? First of all, so we say genetics is a branch of biology that deals with the study of genes. Okay, genetics is the branch of biology that deals with the study of genes. The study of genes, how they function, and even how they work. Okay, so that is genetics. Okay, the study of genes is genetics. Okay, and how genes work and how they function. Okay, we also say that genetics is a branch of biology that studies how traits are passed, okay, from parents to offsprings through genes. Okay, it is a branch of biology that studies genes and how they function. It's a branch of biology that studies how traits are transferred or passed, okay, from parents to the offspring. Also, in fact, genetics, we can say it is what is the science of heredity and variation, all right? We also call genetics the science, okay, of what? Of heredity and what? And variation. And Gregor Mendel, all right, is the father of what? Of genetics, because his pea plant what? His pea plant experiment, okay, laid what? A foundation, okay? And then other scientists, all right, were able to what? To build on what? On it when his work were what? Rediscovered, all right, in the early 1900s, okay? And some of those other scientists that worked, okay, on the what? On the field of what? Genetics, okay? That made some other discovery after Gregor Mendel. Well, number one, we have what? Sutton, right? So we have, what's his name? Walter Sutton and Theodore Boveri, okay? This one in the what? 1902 to what? 1903, okay? This was when they jointly worked, okay? They jointly work on what? On genetics. They proposed the chromosome theory of inheritance. The chromosome theory of inheritance, okay? So they said that what? That genes, okay, that are located, okay, on what? On chromosomes, okay, that will find genes on what? On chromosomes, right? So they work on the chromosome theory of inheritance. Please take note of that. Walter Sutton and what? And Theodore Boveri, all right? Walter Sutton died in the year 1916. Theodore Boveri died in the year 1915, I think, okay? So those were what their work, all right? So they worked in the early 1900s, from 1902 to 1903, okay? They work on the chromosome theory of inheritance. Another scientist that worked, okay, on what genetics was what, Thomas Hunt Morgan, okay? Thomas Hunt Morgan. Okay, this one lived from the year 1862 to 1945. Okay, so this one would use the word the Drosophila. He worked with Drosophila what? Melanogaster, okay, the fruit fly, all right? So he worked what? With the fruit fly. He was the one that discovered linkage. He discovered linkage and crossing over, all right? He discovered linkage and what? And crossing over. It was one that proposed that genes, he proposed that genes are linearly arranged on chromosomes. Okay, that is what Thomas Hunt Morgan from the year 1866 to 1945. Okay, worked on what? On the fruit fly, known as what? The Drosophila melanogaster, all right? Discovered linkage and crossing over, and also he proposed that genes that they are linearly arranged, okay, on what? On chromosomes. Then we have James Watson and Francis Crick. James Watson and then Francis Crick, okay? Crick, I think that is depending on the Crick, all right? This one was in the year, I think, 1953, all right? All these ones, they discovered what? The double helix structure of what? Of DNA. All right, they discovered the double helix structure of what? Of the DNA. Okay, they used the result from what? From Franklin's experiment. All right, from his X-ray studies. All right, so the one that discovered the double helix structure of what? Of the DNA. That is what James Watson and Francis Crick. Then we have what? Rosalind Franklin. All right, you heard me mention Franklin just now. Okay, Rosalind Franklin, okay, lived from the year 1920 to 1958, okay? So she was the one that she's a lady, okay? I remember me talking about what? James Watson and Francis Crick developing the DNA model, all right? Using what? images from the X-ray studies of what? Of Rosalind Franklin, okay, of Franklin, all right? So she was one that took the what? The X-ray diffraction images, okay, all right, of the DNA, all right, which enabled what? James Watson and Francis Crick to what? To build the what? The DNA model, all right? So a famous photo was called Photo 51. Photo 51, all right? It was called Photo 51. That what? James Watson and Francis Crick, okay, used to what? To develop the DNA model. They also have Maurice Wilkins. Maurice Wilkins, okay? Maurice Wilkins joined what? James Watson and Francis Crick, okay, to develop the DNA what model. He worked with them, okay? So Maurice Wilkins, all right? He worked with James Watson and Francis Crick, all right? He shared the Nobel Prize. He shared the Nobel Prize with what? With James Watson and Francis Crick. Okay, he shared the Nobel Prize with them because what he helped them in developing the DNA model also. Then also we have Barbara Clinton. Barbara McClintock. Okay, all right, Barbara McClintock, right? This one lived from the year 1902 to the year 1992, all right? He won the Nobel Prize in 1883, all right? He was one that discovered what we call jumping genes. Barbara McClintock discovered what? Jumping genes in maize. All right, he discovered jumping genes in what? In maize. All right, he won the Nobel Prize in the year 1883. Then we have Frederick Griffiths. Okay, Frederick Griffiths. He did the Griffith experiment. All right, we have Frederick Griffith. All right, okay, Frederick Griffiths. All right, do not mind all of these names, okay? Just to make you understand and make not to be caught by JAMB or WAEC or any external exam, okay? Right? They can ask you which of the following scientists did not work on genetics, okay? So if you do not know all of these names, all right, you should just know them, okay? Just know them. If they're not important, I would have told you. All right, so Frederick Griffith, this one 1928. All right, he conducted the Griffith experiment, okay, and with bacteria, all right. His experiment showed what? Transformation, okay. It concluded that genes can be transferred between what? Organisms. Okay, so that is what Frederick Griffith experiment, okay, was one that conducted the Griffith experiment with bacteria, all right, which showed transformation, okay, showed that genes can be transferred between organisms, okay. So please, I was talking about Barbara McClintock here, all right. Sorry, I mean 1983. He won the Nobel Prize in 1983, not 1883. All right, please correct that. Okay, he won the Nobel Prize in the year 1983. Then we have Oswald. We have Oswald. Okay, we have Colin. Okay, then we have Macklin. All right, in the year 1944. All right, they proved, three of them. Okay, they proved that DNA. They proved that DNA, okay, is a genetic material. All right, material. Okay, and not protein. All right, so Oswald Avery, Colin MacLeod, and Macklin McCarthy. Okay, Oswald Avery. Okay, Oswald Avery, Avery. Okay, Colin MacLeod, Macleod, and Macklin McCarthy. All right, in the year 1994, they proved that DNA, all right, is a genetic material. Okay, and the DNA is not a protein, but a genetic material. Then we have Hershey and Chase. Okay, then we have Hershey and Chase. All right, Hershey Hershey and Chase. Okay, sorry, I do not remember their full names. Okay, but Hershey and Chase. All right, they confirmed. Okay, they worked with viruses. So they conducted the Hershey Chase experiment. They named it after them. After themselves, Hershey Chase what experiment in the year 1952. 1952, okay, and they confirmed what Oswald Collins and Maclean, okay, said that DNA is a genetic material. They're the one that confirmed it, okay. They confirmed it in their experiment, all right. They worked with viruses. They used viruses, okay. They confirmed, all right, that DNA is a genetic material. And DNA is a what? It's a genetic what? Material. So, like we said, okay, Gregor Mendel. Let us now talk about what the works of Gregor Mendel. All right, Gregor Mendel, like we said, was a very curious person. All right, a very curious person. He was intelligent and at the same time was curious. All right. So please, we have discovered that many of you do not support us. Okay, do not even like our videos. Okay, you say send us money. You don't say you will send money. All right, so please like the video. Just like the video you are watching. Okay, is this something too much to ask for all of this? No. So please kindly click on the like button. All right, liking the video will make other students easily find these videos. Okay, and because it is helpful to you, it will be helpful to them. It's not as if you like the video. It's not say they'll be paying us if you like the video, all right? It's just for other students to be easily able to find this video. Also, click on the subscribe button, all right? If you have not subscribed to this channel, Knowledge Capsule, click on the subscribe button and follow us, all right? And if you are watching from Facebook, click on the follow button. If you are watching from TikTok, click on the follow button, all right? So please, all this you are doing is to help other people out there. All right. Also, click on the subscribe button if you're on YouTube. Okay. If you have not subscribed to this channel, click on the subscribe button. Then copy the link of this video. All right. Wherever you're watching for, and share it to your friends, to your loved ones. Come and see genetics and variation that we were taught and we do not really understand them. All right. Come and see how this guy taught it. Come and see another method he brought. All right. So please just ensure you are supporting this video all the way you can. Okay. So like we are saying, Mendel was a very curious person, all right, and his curiosity, all right, when he was working in the monastery, all right, made him to work on work using the pea plant. The pea plant, okay, called Pisum sativum, all right. So he worked on the pea plant, known as as the Pisum sativum, okay, and then it made him to draw up an understanding, okay, devise understanding to know how traits, okay, how genes were transferred, okay, from parent to offspring, and then from then was able to propose two laws. Okay, proposed two laws: the law of segregation and the law of independent assortment. Okay, and even aside from these, he went ahead to develop the monohybrid cross. Okay, monohybrid cross, and then also the dihybrid cross as well. All right. So these were all the works of Mendel. But then, as a curious student, also you will ask, why did Mendel choose a pea plant? Why did he choose the Pisum sativum? Okay, but then other plants. Why was it that it was only pea plants he was able to use to conduct his experiments? Okay, and the reasons why he chose the pea plant are not far-fetched. One of the reasons why he chose a pea plant is because, number one, pea plant has a very short lifespan. Okay, so when you want to conduct an experiment, okay, you should not take a species that will take maybe years, okay, to grow. Okay, imagine you taking a cashew tree, all right, or taking a mango tree, okay, or what is it called? Any tree, okay, that will take a long time to grow. All right, so you have to take a plant that can easily mature within a short period. All right, so that means that Mendel It was from recorded from 1822 to 1884. Okay, imagine what it took a plant that would take maybe 20 years to mature, right? To see that it doesn't make any sense. All right. So if you want to conduct an experiment, you choose a plant, all right, that has a very short lifespan. And of course, they were beneficial. Pea plants were beneficial, all right, to what? To the experiment it was conducting, all right. They had a very short lifespan. Number two, okay, the pea plant is what? It's bisexual, all right. It's bisexual. You know what I mean by bisexual? Okay, it has both what? The male and female what? Reproductive what? Organs. All right, it has both the male and the female reproductive organ, and that means that, okay, even without cross pollination, all right, you can what? It can carry out self what? Pollination. All right, so it was what? A bisexual plant. So you are seeing some of the reasons why Mendel chose what? The pea plant. Okay, it was bisexual. It had both the male and the female reproductive what? Organs. Okay, in it, that means it can what? Self-pollinate. It doesn't even need to go and what? Find another pea plant to what? To be crossing it with. All right, so they can self-pollinate. It could breed the same type of plants. Okay, and as a result, it will give him what? Pure breeds. Okay, all right, it could breed the same type of what? Of plant, and as a result, you will be able to what? To get pure breeds. And pure breeds, okay, are the condition where it can get what? Characters. Where it can get characters. It can get characters in homozygous conditions. All right, so that was part of the reason why what he chose the pea plant was bisexual. All right, it could self-pollinate. Okay, it could give him what? Pure breeds where it can get characters in homozygous or condition. Also, it can what? Cross-pollinate. Okay, it can cross-pollinate. Pollinate. Okay, cross-pollination will make him to have what? Cross breeds. Cross what? Breeds. Okay, self-pollination will make it to have pure breeds. Okay, while what? Cross-pollination will enable you to have what? Cross breeds. And cross breeds is when what? Mixing of characters. All right, in cross breed, we are what? Mixing characters. Okay, in cross breeds, mixing of character is what is taking place, and you can get what? Plants, or you can get characters in what? In heterozygous conditions. Now, heterozygous conditions. All right, so that's why what it was choosing the pea plant. Bisexual could cross-pollinate also to get cross breeds, okay, which involve the mixing of character to get what? Characters in what? In heterozygous what? Conditions. And along with all of these, it will be easy to understand because they all add what? Contrasting what? Characters. Contrasting characters. All right, so Mendel's reason for choosing a pea plant is not far-fetched. Okay, so this is why he chose what? The pea plant. All right, they are contrasting characters. Pea plants have 7 pairs. Pea plants, they have 7 pairs of contrasting characters. Okay, so you can see why he chose what? The pea plant. All right, pea plants, they have what? 7 pairs. 7 pairs of contrasting characters. All right, so do you know what it means for a plant to have 7 pairs of contrasting characters? All right, so this is why, these are some of the reasons why the pea plant was chosen by Johann Gregor Mendel in the conduction of his experiments. So now, all right, what are these 7 pairs of contrasting characters that we have? Number 1, we have height. Okay, so the height, all right, we're going to looking at what the dominant trait. I'm going to look at the recessive trait. Okay, so dominant. Okay, here, recessive. Okay, so for the height, we have the tall. Okay, for the recessive, we have the short. Okay, so the height was one of the characters. Okay, one of the contrasting characters that the pea plant had. Okay, also the seed shape. All right, also have the seed shape. Okay, let me just name what these characters are. We have the seed color, we have the flower color. All right, will I have time? Will I have space to write all of them out? Okay, 1, 2, 3, for flower color. All right, we also have pod shape. All right, this is 5. Then we have pod color. Then this is number 6. Then we also have flower position. Flower position. All right. So for the height, okay, we have what the dominant trait is what tall, the recessive is what is short. Okay. Now for the seed shape, okay, we have what for the dominant we have round. For the recessive we have what wrinkled. Okay. Then for the seed color we have what yellow for the dominant. Then we have what green. Okay. For the what for the recessive. All right. So we have yellow color for the seed color. For the dominant trait, then for the recessive, we have what? Green. All right. Then now for the flower color, we have what? Violet. Then what for the recessive, we have white. Okay. Then for the pod shape, we have what? Inflated for the what? For the dominant. Inflated. It has an inflated pod shape. While for the what? For the recessive, it has a constricted what? Pod what shape. Then also now for the pod color we have what? Green. Okay, while this one is what? Is yellow. All right, yellow. Then what for the flower position we have for the dominant we have what? Axial. Okay, while what for the what's it called now? For the recessive we have what? Terminal. All right, so these are what the all of the contrasting character that the P plant had. It had 7 pairs of contrasting what characters: the height, the seed shape, the seed color, the flower color, the pod shape, the pod color, the flower position. All right, so that means Mendel could have taken any of these what characters, okay, to do is what he studies. All right, so he had many options. Okay, it had 7 pairs of contrasting characters. So it was very easy for Mendel to observe the physical differences. The physical differences, okay, that were what? That were present what? In the P plant, okay? All these things that are what you can see, okay? You can see them with your eyes, all right? So Mendel could observe all of these physical differences with what? With his own eyes. So these are some of the reasons why what Mendel picked what? The P plant, all right? Short lifespan, contrasting characters, bisexual nature of what? Of the pea plant. Okay, so it could self-pollinate, it could cross-pollinate. Okay, these are the reasons why what Mendel chose the pea plant. Then of course we knew that what Mendel performed some experiments. Okay, so in this experiment, what did Mendel do? How did he perform this experiment? How did he perform this experiment? Please ensure you have liked this video, subscribe to this channel, and share the link of this video to your friends and loved ones. So the first thing Mendel did was what? Was to pick what? A particular character. Okay, so among all of the 7 contrasting characters, he picked what? A particular character. Okay, it could just be what? Only height. All right, could pick what? Could pick what? The seed shape. Could pick what? Seed color. Could pick what? Pod color. Could pick flower position. But it took only one what? He picked only what? A particular what? Character. All right, and then what? It took what? 2 plants. All right, 1 character. Okay, 2 plants. All right, it is this one that he named the what? The monohybrid cross. Okay, so in monohybrid cross, mono means 1. Please, so you never confuse monohybrid with dihybrid. Okay, mono means 1, di means what? 2. Okay, so in monohybrid cross, it took what? 1 character, 2 plants. Okay, so it's the mono and di is always in respect to the character is what that was taken. All right. So in monohybrid cross, it took 1 character with 2 plants. In dihybrid, it took what? 2 characters. Okay, it took what? 2 characters. All right. So never confuse monohybrid question with dihybrid question. All right. So it took what? 1 character and took what? 2 plants, and he named it what? The monohybrid cross. So in the case of monohybrid cross, okay, it took what? Only what? 1 character, only 1 character, and then it took 2 plants. Okay, so what did he now do after picking what? 1 character? Let's say for example he picked what? The height. You pick what? The height as a character. Okay, you know that height has what? 2 alleles, isn't it? Whether be what? Be tall, okay, or what? Or short, or small, or dwarf. Okay, so it took what? The height. Okay, the height could either be what? Tall or dwarf. All right, so it picked what? A pure tall plant. It took a what? A pure tall plant. So the phenotype for this particular plant is what? Is tall. Okay, that means you can from you know what phenotype is now. We'll explain what phenotype and genotype. Phenotype are the physical observable characteristics that you can see. Okay, just like I'm showing you now this mark and I can see that that's what a green cover. You can see it with your eye. All right. So we do not know the genotype. You may not know the chemicals that we used to make these colors or that was able to make this thing become green like this. All right. You may not know it. Okay. But you can see with your eye. This is phenotype. Those are the one you can see. All right. You know that behind, okay, there are some things, so all right, that may be there, but you see this one you are seeing is the phenotype. They are the observable physical characteristics that you can see. All right. So the genotype, the phenotype here is what? Is pure tall. So you are seeing it is pure tall. So it took what? A pure tall plant and it took what? A pure dwarf plant. Dwarf plant. All right. So now, I already told you that if you are taking pure tall plants, okay, if you are taking a pure tall plant, the genotype will be TT. It is pure. So it has to be homozygous, okay? It's a pure breed, okay? Why what? For the pure dwarf plant now, to be what? Small t, homozygous. It's a pure, since it's pure dwarf plant, okay? So it took a pure tall plant. That's why I see capital letter T, capital letter T. Then he took what? A pure dwarf plant. That's why I see what? Small letter t, small letter t. So please do not be confused. Whenever you hear pure tall, okay, we're talking about what? A plant in the what? In the homozygous or condition, all right? So since it's pure tall, TT, capital letter T, pure dwarf, small letter t, small letter t. So now let me make this clearer, okay? Let me make this clearer. Let's make this clearer for you to see. All right. So phenotype. Okay. Then genotype. Okay. The gametes. All right. So have the phenotype. Right? Genotype. We have the gametes. So the phenotype here is what? Is tall plant. It's a tall plant. And it's a pure tall plant, okay? Why this side is what? It's a pure dwarf plant. Okay, so that's what we are seeing on the outside. That this one is pure tall, is pure dwarf. So on the inside, the genotype will be what now? TT, okay? Why what? The genotype for this will be what? TT, okay? So this is pure tall, this is pure what? Dwarf, okay? So now, the gametes. I taught you what in mitosis and meiosis, okay, especially meiosis, all right, that's what the gamete will only have the number of chromosomes, okay, like for example, if we have 46 chromosomes, okay, our sex cells, okay, we have half of this number, which is 23. I told you, right? All right, that means that that the gamete here, one will come from the parent, one will come from what? One will come from the father, one will come from this pure tall, one come from this dwarf or plant, okay? So that will not be the gamete. So it will end up having half the number of chromosomes. So this means that now? Our gamete here, for this top plant, our gamete here, this one will bring 1, okay? It will have half of it, okay? Then for the dwarf plant, it's have this, okay? So now, in our F1 generation, generation, this one is called the first filial generation, all right? So in our F1 generation, okay, if they combine, all right, this will combine with this, okay, what I want to have? I want to have TT, all right? So this is what I'm going to have. This one is the what now? This one is what? Is the genotype, okay? Now, but is this homozygous or heterozygous? First of all, let me ask you this question. Is this a tall plant or a short plant? Okay? I told you about dominant and recessive alleles, okay? So now, the tall is dominant over the short, over the dwarf, all right? Okay, that means that this plant, okay, will be a tall plant. To be a tall plant. That means phenotypically, what we go to see with our eye, and I say this plant is a tall plant outside, okay? But the genotype, okay, is not a pure tall plant. It's not an homozygous tall plant. Instead, it is an heterozygous, okay, tall plant. All right, because you can see that these two alleles, they are dissimilar. They are dissimilar. they are not the same, okay? If they were to be the same, TT, okay, or let me say, sorry, TT, this one will have what? Homozygous what? Tall plants, okay? But since it's what? T and small letter T, it's a tall plant, okay? Because the T, this capital letter T is dominant over what? This T, it is masking, it will mask what the effect of what? This recessive what? Allele, all right? So I want to have what? A heterozygous what? Tall plant, because what? The alleles are what? Are dissimilar. I hope that is very, very clear. So initially, we had what? A pairing, which was what? Was similar. We had a pairing which was similar. But what was our result in the first filial generation? Okay, we are getting a pairing that is what? That is dissimilar. That is not the same. Okay, that's heterozygous. We began with what? It's homozygous. All right. Was homozygous were pure tall, pure dwarf, okay, but we are getting an heterozygous or tall plant, okay. So this is what made what Mendel to be more curious, okay. Why are we having this, okay? So now, what did Mendel do next? Please, when you are crossing, most time, please ensure you make use of what we call a Punnett Square. A Punnett Square, all right? So many of you, you know what the Punnett Square is, but you do not know it's called a Punnett Square, all right? A Punnett Square, all right? When you do something like this, okay? You do like this, you draw table, they'll say draw table, all right? You do like this, okay? You now say here, T, yeah, T, okay? You now say T times T, T times T, T times T, T times T, okay? So in the first filial generation, what do you observe? You see that what? All of the plants, okay, they are pure tall. This is what we did, all right? Then I was taking what? This T, all right? We'll cross them. All of them, they are what? They are all TT, capital letter T and small letter. This is a Punnett square, okay? So let me quickly explain what the Punnett square is for you, okay? So before we continue. All right, so please ensure you have liked this video, you have subscribed to this channel, and of course, you are sharing the link of this video to your friends and loved ones. All right, this is the only way you can help us grow the Punnett Square. So the Punnett Square is a simple box chart that scientists use to predict what possible traits, that is the genotype and the phenotype ratio whenever two parents are crossed. Okay, so we can say a Punnett Square is like a simple box chart scientists use to predict possible traits. That is what now? Your phenotype and genotypes, all right, of offsprings when two parents are crossed, all right? And of course, that is what the Punnett Square is, a simple box chart I showed you, all right? That scientists use to predict what possible traits, that is the phenotype and the genotype, okay? Offsprings when two parents are crossed, all right? That is the Punnett Square. It was developed by Reginald Punnett, okay? Developed by Reginald Punnett, okay, in the year 1905, all right? So Reginald Punnett, all right, developed Punnett Square, all right, in the year 1905. So just think of the Punnett Square like a genetics calculator, okay? Think of it like a what? Like a genetics calculator, all right? What you do is that you write the alleles from one parent on the top, all right? You write the alleles, number one. You write the alleles from one parent at the top, okay? Just like I was showing you, right? When you have a box like this, all right? You have something like this. Then you have like this, you have like this. You write the alleles from one parent at the top, okay? Then you write the alleles from the other parent at the side here. You see? Then you fill in the boxes, okay? You fill in the boxes, TT, okay? TT, fill in what the box is. This is an example, all right? All right, you fill in what the box is, all right? Let's say this is what, this is TT. Okay, let me just say this is what TT. Okay, TT, all right? So you have TT, you have TT, you have what TT, you have what TT, all right? So now, you write what the alleles from one parent at the top, which you are seeing here. Then you write the alleles from the other parents at the side, all right? Then what do you do? All right, you now what see all the possible combination for the for the offsprings or for the children. So that's what basically what the Punnett Square is. Okay, that's what it is. All right. So now let us do an example. Let's say we have what a tall plant. Okay, tall versus short pea plant, for example. All right. Let's say we have a tall versus a short pea plant. Okay, we know that what that T stands for what? Capital letter T stands for what? For tall. Okay, and it is what? It is the dominant what? Trait. Okay, then we now have small letter T. Okay, which stands for what now? For short. This one is what? Is the recessive. Okay, so we know this already. Okay, so now parents cross. Okay, parents cross. What now? TT. Okay, times what? TT. All right, so that is what the parent cross will now be. So now let us cross it. Okay, so that's the parent cross. Let's do this. All right. Okay, so we're going to have what? TT, TT. Okay, T times T is what? TT. T times T is what? TT. T times T is what? TT. Always write the dominant first before the recessive, all right? So you write capital letter T first, even if it's T that's multiplying this T. So it is wrong to say TT like this, all right? So please, I'm showing you how to use the Punnett Square when I so use what? TT. Then have what now? TT. Okay, like we said, it makes us to what? To see what the genotype and what the phenotype of ratio, all right? So now for the genotype, what are we seeing? We are seeing what now? TT, which is tall. Okay, sorry, first of all, I've seen TT, which is still tall. Then I've seen TT. Then I've seen what now? TT. All right. So the genotype ratio, all right, will be what now? 3:1. All right. Why? Because we are seeing TT. This capital letter TT is for what? Is for tall, isn't it? But homozygous tall. Then this capital letter T, small letter T is still what? Tall. But it's heterozygous or tall because the two alleles here are similar, isn't it? This TT2 is also what? Heterozygous tall, all right? So they will now have what? TT, which is what? A pure short plant, all right? So the genotype ratio will be what? 3 tall and what? 1 short, isn't it? 3 tall and 1 short. That would be what? The genotype what ratio, all right? 3 tall plants, then 1 pure short plant, all right? But sorry, this will not be the genotype ratio, rather. Sorry, that's an error. Okay, so the genotype ratio will be showing us what? It will be showing us 1 pure tall plant. Sorry, 1 pure tall plant. Sorry, I'm very, very sorry. Okay. Oh, this momomi. It's 1 pure tall plant, then two heterozygous tall, then 1 pure short plant. This will be the genotype. On the genotype level, this is what we are going to be seeing. All right. So inside the genotype, which is not visible to our eyes, this is what to be showing. It will be showing 1 very pure tall plant, 2 heterozygous tall plant, and 1 what? Pure short plant. Why what now? What's it called? The phenotype ratio. So on the outside, which are the observable physical characteristics? What are we going to be seeing? Wait till you're concerned, and I be say 3 plants tall, isn't it? So 3:1. So I think I was trying to mix both of them together before, right? So for the genotype ratio, so it tells what the Punnett Square shows what the genotype ratio, which was 1:2:1. Why the phenotype ratio will be what now? 3:1. Okay, 3 tall and 1 short. Okay, so that is basically all about what the Punnett Square. Okay, so in what ways are the Punnett Square useful? All right, so number 1, it helps predict traits. The Punnett Square help to what? To predict what? Traits in offsprings. All right, it helps to predict traits. Number 1, usefulness of the Punnett Square. Number 1, it helps to predict traits in offsprings, okay, or in the children, right? So it helps us to predict what traits in offspring. Then number two, it's what shows dominant. It shows what dominant and recessive inheritance. All right. So the Punnett Square also helps to show dominant and recessive inheritance. Okay. And then lastly, number three, it's used in genetics. It is used in genetics problems for both plants and animals and even humans. Okay, so these are the usefulness of what? Or the Punnett Square. It helps to predict traits in offsprings, shows dominant and recessive inheritance, used in genetics problem for both plant and animals, as well as even what? Humans. Okay, so please take note of all of that. So that's the Punnett Square. All right, I've done a full class on Punnett Square just for you to understand Punnett Square very well. All right, so now we have to now continue with with our Mendel experiment. So in the first filial generation, what did we get? We got what all heterozygous tall. All right, we got all heterozygous tall. If you use the Punnett Square, all right, for the first generation, all right, what do you see? This is the Punnett Square now for Mendel. All right, T T T T 1T 1T. All right, so we crossed. Value of pure tall plants. All right, so now let us see the Punnett Square to do the cross. All right, so we have what? T, okay, T, then we have what? T, T, okay, so now T times T is T, T, all right, T times T is what also? Sorry, T times T, okay, T, T, T times T, 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 T times T, 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 T times T, T, okay, so they were all what? Tall, all right, in the first generation. They were all tall, but although they were what? Heterozygous what? Tall. All right, so it is the Punnett Square that said us what to know all of this now, all right? So they were all heterozygous what? Tall. So the genotype here, genotype, okay, will be what now? T, okay, this is genotype TT, 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 and what? TT, 1, 2, 3, 4. All right, this is genotype. All right, that on the inside, or the one we cannot see, is what it's showing us. All right, but the one we can see is that what? Which is the phenotype now? The phenotype is that what? All of them are what? All of them are tall. All right, that's what we'll be seeing outside. It does not concern whether it's TT now on the outside or it's what? TT. Okay, but we are seeing tall plants. Now, it just concern us with that, okay? For what? For phenotype, okay? That is the observable one you can see with your own eyes. You had what? One parent that was what? That was pure tall. Had another one that's what? Pure short, okay? But at the end, okay, of what? The first generation, you are seeing that what? All of them are what? Are tall. Are tall, all right? The tall what? The tall what? The pure tall dominance, okay, showing itself, is expressing itself, is maxing the effect of what? Of the short one, okay? Within the first generation, we were not even seeing any short plants. All of them just tall, okay? Which made this word very, very interesting, okay? All right? So that is what the first generation, all right? They were all what? Pure tall plants. This is the Mendelian idea, Mendelian. Okay, it is what? The Mendelian what? Idea. All right, it's called what? The law of what? Of dominance. All right, the law of what? Of dominance, which says that in heterozygous conditions, okay, the factor or gene which expresses itself physically, okay, which expresses itself physically is known as what? As the dominant, okay, while the other one, which is unexpressed, all right, is known as what? As the recessive. So Mendel's law of dominance, all right, like we are saying it now. Mendel's law of what? Of dominance. Please, if you want to write this, you write it, okay? If you want to write, you write. Right, so Mendel's law of dominance said that what? In heterozygous condition, Okay, as you can see, all of them here, they are what? They are in heterozygous what condition, all right? So this is where his first law of dominance came from, all right? So that in heterozygous condition, the factor or gene which expresses itself, what itself physically, okay, is what is known as the dominant allele. While the other which is unexpressed, is known as the recessive allele. All right. So Mendel's law of dominance. Okay, he said that in heterozygous condition, all right, the factor or gene which expresses itself physically is known as the dominant allele, while the other, which is unexpressed, is known as what? As the recessive allele. So now, what did Mendel do? Okay, he said to himself, so in the first generation, I took what? A pure tall plant, I took a pure short plant, and then across them, they gave me all tall. All of them were tall. Okay, so why not? Why should I not just take this plant in my F1, okay, and cross them? Okay, why don't I not take two plants, okay, from what? This is my F1, all right? Remember, these are what? 4, okay? Why not take two and cross them and see? All right, so this one's now become what? Parents now. Remember, they were initially what? Offspring, okay? He now made them what? Parents, okay, in the second generation. So that was what Mendel did, okay? So now, he was curious to see what will happen, all right? So he now said, let me take two, okay? This was two parents now, okay? Two plants, okay? From the F1 and cross them together. So he did what? The Punnett Square. So now, your gametes here, okay, will be what now? T, all right? T, all right? So now, we'll do our Punnett Square and see what that will give us. And see what this will give us. You already know everything about the Punnett Square now and what it helps us achieve. All right, so let us do a Punnett Square and let us see. All right, so do not mind my diagram. All right, so now it took what is T, isn't it? And what? T. Okay, now, so you know how to use a Punnett Square. This time, this is what? T, okay? Then this times, this is what? Is T. Then this times, this is what? T. Okay, but like I told you, don't write the recessive first before the dominant. Okay, so you can clean this now and say what? T. Then this time, this also is what? Is T. All right? So now we have done our Punnett Square. In our Punnett Square, okay, we have what? Cross what? Two heterozygous what? Tall. Okay, we cross two heterozygous what? Tall plants. Okay, which is what I want now, TT. Okay, we took two and then we crossed them. So what do we observe, all right? So from our Punnett Square, what we get? Okay, what do we see? We see that what now? We have what? How many tall plants are here? 1, 2, 3. TT is tall plant, isn't it? TT is not a tall plant again. TT is not a tall plant again. Then now have TT, which is what? Short plant. Okay, so now what is this showing us? We're having 3 tall plants. Then 1 what? Dwarf plant. Okay, so this is the result of what? Of our crossing. Okay, when we made what? The product of the first generation, okay, the parents, and we cross them. So we self-pollinate them. Okay, we self-pollinate them, all right? So this is what the result. We had 3 tall plants and 1 dwarf plant. We got what now? 1 pure tall plant, 1 pure tall, 2 heterozygous, okay, 2 heterozygous were tall, okay, that is this and this. Then we have what? 1 dwarf plant, 1 pure dwarf what? Plant, okay? So this was the result, okay, of what? For our Punnett Square, all right? So in the first what? From the beginning of the experiment, okay, we crossed 1 pure tall and 1 pure short, okay? The result was that we got 4 heterozygous what? tall. All right, we made what those what heterozygous tall, made them parents. We cross them again. All right, what do we observe? We observe that what now we are now having what one pure tall again. Okay, two heterozygous were tall, and then one short. Okay, one pure short also. All right, so this was pure tall and this pure short that disappeared in the first filial generation. All right, they have reappeared again in the second filial generation. We now even had a character which was not even there at the beginning, which is what? This heterozygous tall, because we did not begin with what? With heterozygous tall at all. All right, the only thing we began with was this and what? And this. So we now have what? A new what? Character. Isn't it? As you can see, we're having two heterozygous tall plants in the second filial generation. So in this case, what do we observe? We observe that what? There has been what? Segregation of characters. Okay, so there has now been what to call what? Separation of what? Of characters. Segregation of characters has happened. So we see that the alleles that were there, okay, or rather the alleles that were coding, okay, for that particular character has segregated. Okay, it has what? Separated. It's segregated during the formation of gametes, and then even during fusion, it's again what? Segregated also. But in the second filial generation, we see that again it has come back. Okay, so segregation has what? Has happened. All right, so that is what you see. You see the first filial generation, all right, they segregated, they separated. All right, but then during fusion, okay, segregation still happened, right? But again, you have seen that what? Segregation has happened again, okay, because it has now what? Come back, right? In the second filial generation. And this is what we understand as the law of what? The law of what? Of segregation. All right, this is what we understand as the law of what? Of segregation. And in segregation, the two alleles, okay, the two alleles, okay, for what? For that character, we segregate during what? Gamete formation. They will segregate. All right, they will segregate. The two alleles will segregate during what? Gamete formation. But please note that what? Both parents, okay, they contribute equally. Okay, they contribute what? Equally to this what? To this said segregation. That's what that is happening. So now let me explain the law of segregation in a way that what you may maybe understand. Okay, if you do not get what I've been saying, right, I think this will help you have a better understanding about the law of segregation. All right, please like this video, subscribe to this channel, and share to your friends and loved one. All right, so law of segregation, that's Mendelian law of segregation. All right, now imagine you have a bag. You now, I'm talking to you that are watching this video now, right? You have a bag with two candies inside the bag. Okay, imagine you that is watching this video so that you understand law of segregation very well. All right, you have what? A bag with two candies inside the bag. Okay, now one is what? Is a red candy. One is red, right? Let's call this one the dominant one. Okay, all right, then the other one, which is the second candy now, right? Let's say is what? Is blue. All right, this one is what? Let's call it the recessive one. Please follow the story one after the other. All right, you have two candies inside your bag. You that are watching this video, you have two candies inside your bag. One is red, which is the dominant one. Then the other one is blue, which is the recessive one. Now, when it's time to share candies, okay? When it is time to share candies. All right, share candies here. All right, you use it to mean what? Making babies. All right, so when you want to make babies, all right, you give what? You can only give among these two candies that you have, you can only give what? Only one out. All right, so you give one candy out. It may be the red one or the blue one. All right, you give what? Only one candy out. And then what will happen? The other parent, all right, will give what? One candy also, isn't it? All right, so when you want to make babies, all right, what happens is that what now? you will give one candy, you bring one candy, because what both parents must contribute to the offspring, isn't it? All right. So you are bringing one candy, you are giving one candy out to the child. The other parent also is giving one candy out. So now what do you observe? Okay, you observe that now the child now will end up with how many candies now? Will he have 4 candies? No. Okay, the child will not have 4 candies, but 2 candies, because you had 2, the other parent have 2. You gave 1, the other parent gave 1. 1 plus 1 is what? Is 2. So the child will end up with 2 candies again. All right, which is the original one that you had. Okay, you had 2, you gave 1. The other parent had to give 1. All right, the child now has 2, which is now like the 2 candies that you had before. All right. So what do you observe here? All right. The child now has 2 candies. What this means in science is that every parent has 2 versions. Okay, has 2 versions, or what we call alleles. Okay. All right. Every parent has 2 versions, or has 2 alleles of a particular trait. Okay, every parent has 2 versions, or alleles of a trait. All right. So now, when you are making eggs, or you are making sperm, the 2 versions, they separate. Okay, the 2 versions we are talking about here, they separate, and as they separate, one goes to each gamete. All right, one goes to each gamete. All right, so the 2 versions, or the alleles of that trait, we separate. One will go to each gamete, and when fertilization happens, the child will get 1 from the dad, 1 from dad. Okay, you get 1 from mom also. Okay, so this is what happens basically. Now let us see an example. Okay, just like we're using the example of tall and short pea plant. Okay, let us see an example using the tall and the short pea plant. All right, tall versus short pea plant. Okay, what do we notice? We know that T is for is for tall, which is the dominant one. Okay, then small t is is for the short, which is the recessive one. Then of course, after the first generation, all right, first filial generation, we had the parents now became TT times TT. All right. So now each parent will give one one allele out. Each parent cannot give 2. Okay, you cannot give 2. All right. So, and at the end, what do we observe? Okay, if you do a Punnett Square, okay, we observe that what happened? This is TT. Okay, sorry. Let me say it like this. All right, TT. We have TT. We have TT. We have TT. We have TT. All right. This is what we got as the result. Each parent will give this T, or will give this T, but they cannot give both. Okay, they cannot give both. So at the end, what do you observe? We observe that now the child. Okay, had we have 3 tall children. Okay, 3 tall. Okay, and one short. This is what we observe. This is what we see. Okay, at least this is what the phenotype is telling us. So the baby could be Could be tall. Could be tall, which is TT tall. The baby could be Also this TT tall. Okay, this genotype TT. Okay, TT tall. Okay, or the baby could be Could be short. All right, TT. Okay, this one is short. All right, this is what we are seeing. Okay, so the baby could be Tall. Could be TT tall, or pure homozygous tall, heterozygous tall, or pure short. All right, so this is what we are seeing. So in the end, the traits, what happens is that they separate. Okay, they mix, and then they come back again. They come back again, just like like candies, okay, in the bag, just like the illustration I'll be giving to you. Okay, that is what we call as the law of segregation. So Mendel's law of segregation is saying that each trait is controlled, okay, by by factors, okay, which we know as as alleles, which we know as as alleles. Each trait is controlled by a factor, okay, which we call was, which is called what? Alleles. And that these two alleles separate during what? Gametes formation, all right? That's what each trait is controlled by a factor, which we call alleles, and that these two alleles, they separate during what? Gamete formation. And what happened? Each gamete gets what? One what? Allele, all right? So that's what the law of segregation is saying. Each gamete we got, the two alleles will separate during gamete formation. Each gamete will get what? One allele. And at fertilization, the alleles, now at fertilization, the alleles will what? Come back together again. So at fertilization, the alleles will then what? Come back together, giving the offspring two alleles, okay, for what? Each trait, all right? One from each what? Parent. All right, so that is what? The law of segregation. Each trait is controlled by a factor, which is called alleles. These two alleles, they separate during gamete formation. All right, each gamete gets one allele. All right, so these two alleles, they separate during gamete formation. That is what? The egg and what? The sperm. All right, so each gamete will get one allele, and at fertilization, the alleles will come back together again, giving the offspring two alleles for each trait, one from each parent. So at the end of what? Of monohybrid cross, we get what? 3 tall plants, and then we have a one what? Short plant, okay? But then of course, using the Punnett Square, we know that what? We also have what? The phenotype and the genotype ratio. So let us please see that phenotype and genotype ratio, all right? So what's the phenotype ratio at the end of what? Monohybrid cross, all right? So let us see. All right, because at the end of monohybrid cross, we have what? One pure tall plant. We have two heterozygous tall plant. Then have what? One pure short plant. Okay, so we have 3 tall plants in total. 1, 2, 3. Then have a one short plant. Okay, at least this is what we are seeing. Okay, so the phenotype ratio. Okay, the observable what? Characteristic. So the one where we go to see for outside and say 3 of this plant, they're tall, 1 short. Okay, so I'm going to have 3 tall, ratio 1 short. Okay, so that 3 to 1. Okay, that's what we're going to have. At the end, so you may be, this is a question that you'll be asked in the exam. Okay, that at the end of what? Monohybrid cross. Okay, Mendel had what? What's the ratio? What the phenotypic what ratio? All right, it's 3 to 1. 3 tall, 1 short. Okay, but the genotype, what are we going to be seeing inside? I'm going to see 1 pure tall plant, 2 heterozygous tall plants, and then what? 1 pure short plant, right? So this is what we're going to be seeing. This is what the genotype ratio now, right? So this is the phenotype ratio, and this is what the genotype ratio at the end of what? Of monohybrid cross. So some of you will say that, why am I always using the Punnett Square? Can I just not cross the plant? All right, using another method? Yes, you can use another method, but the Punnett Square makes it very easy for you. All right, so you know that the parents in the second generation, parents TT. All right, that's a TT. Okay, so you can cross like this. Let me show you a simple way to do it. Okay, if you want to do this method, all right, you will, if anyone you are using, you keep the other one constant. Okay, if I want to use this one to cross this one, okay, this one will be constant. So you carry this one like this. You go and meet what? This one like this. Okay, so this one will give us TT. Abi, capital letter T times capital letter T will give us this TT. That is 1. Then this one is still constant, so you use to cross the other one too. All right, so let's go cross. So I'm going to have what? T, capital letter T, and what? And small letter T. All right, so now you now begin with this one now. Okay, so this one will cross with this. Okay, so this capital letter T and small letter T, so TT. You see it, bar? Then also this one is still constant. You use this one now to now cross this one, which is the one we have not crossed. Okay, so you cross it. All right, Any way you want to do it, okay, whether you are bringing it here or you are bringing it that way, okay, let us take it this way, okay? Capital letter T and, sorry, small letter T and small letter T. You cross it, so I'm going to have what? T, T, all right? I hope this is very, very clear. Okay, so you see the same result I'm going to get if you use the Punnett square. So for this method, what you do basically that you keep, if you are using one to cross the other one, you keep the other one constant. So hold this one constant, use this one to cross this two. Then hold this one constant, use this one to cross this two. So it's not the same result we're getting. Okay, we're getting TT, 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 and TT. Okay, so it's almost what? Exactly the same result. So not almost, is exactly the same result, but you must know how to what? Do this method. Okay. Also, one of the most important things to take note of, all right, in this monohybrid cross is what is the phenotypic and what and genotypic ratio. We are saying that what are the genotype ratio is what is 1:2:1. 1 pure tall, 2 heterozygous tall, then 1 pure short. Then phenotype ratio or phenotypic ratio, okay, is what is 3:1. All right. So now, but how are these traits getting what expressed? All right. We have seen that what this is the ratio in which what they appear. But how are the traits being what expressed? So now, I've told you that we have the genotype ratio, we have the phenotype ratio, and then we have a gene, okay, a gene that obviously that is coding for what? Height, because the character we have taken, all right, is what is height, all right, is what we considered, all right. So now we have a gene that is coding for height, maybe pulse color, it may be seed shape, maybe anything, okay. We have a gene that is coding for any of those characters, okay. A gene is coding what? For height, okay? So how exactly is this gene doing it, all right? How is this being expressed? So now we have what? A gene, all right? We have a gene, okay? All right, we have a DNA, all right? Then we now have what? A sequence or a segment of that DNA, all right? That is what? Coding for this height that we're talking about, right? So something like this, all right? Like this, okay? All right? All right, we have a sequence, okay, or a gene that is coding for this our height that we are talking about. All right, that is coding for what? For this height. All right. So how is it doing it? Okay, basically what it does is that it produces proteins. All right, it produces proteins first. It will code the gene here will code. It will code for what? For some certain proteins. It will code for what? For some certain proteins. So this gene now, we have what? We have the genetic information. All right, it have the genetic information, and once it has the genetic information, it's coding for a certain protein to code for this character, which is height. All right, so it will have access to the genetic information, and as it's coding for this height, all right, it does not just go and tell the plant that plant, okay, I'm coding for height, so what you go do that you go tall. No, that is not exactly how it happens. All right, so it has the genetic information already, so it will code for certain proteins, all right, that code for this character, which is the height we are talking about. Then the proteins, okay, will communicate. These proteins here, they will communicate, okay, with each other. The protein that it has coded, all right, the certain proteins that has coded, they will communicate with each other. These proteins could be in the form of hormones, okay, like for example, growth hormones, all right. So they communicate with each other, all right. So these proteins communicate with each other, all right. They'll be in form of hormones. For example, growth hormones. Like in plants now, we have auxins, all right. That's why when we did endocrine system, okay, we talk about chemical coordination in plant also, okay. So like the auxins in the plant also, and it will be producing certain enzyme that will trigger this growth. Okay, it will produce certain enzymes, okay, that will trigger the growth that we're talking about. So this height that it's coding for, this is what happens. It codes for certain proteins. The proteins communicate with each other. All right. The genes already have the genetic information. All right. So it happens like it produces hormones, like growth hormones. Like for the plants now, like we said, the auxins. Okay. So After this now, to produce what? Some enzymes, okay, that what now enables what? The plant to what? To grow. So when I say that what? That gene code for a particular character, okay, what it does basically is to what? Is to what? To code for what? For certain proteins, okay, which is now in turn what? Produces what? Some hormones, okay, in form of what? Of enzymes, all right, which now what? Trigger what? The growth we are talking about. So when we have what? A gene, all right, when we have a gene coding for a particular character, all right, first of all, it will what? It will code for certain proteins. First of all, code for what? For certain proteins, all right, then for the proteins, it will be in form of what? Of hormones or what? Enzymes, which will now in turn what? Trigger growth. Okay, so that is all about what Mendel's law, all right, and what monohybrid cross. Okay, so we'll still have law of independent assortment. Okay, we're going to look at that law when we talk about dihybrid cross. Okay, so please just what? Stay put. So now we're going to go to the O3 Schools app, answer questions. We're taking a lot of time, and then we'll be out of here. All right, so let us go. This question will be very easy now. So to answer them will be very easy. You'll be able to answer them by yourself. Okay, so I don't go to see a few. You go to the O3 Schools app. I'm going to search for more questions so that you gain confidence. If you cannot answer questions by looking at them after learning from us, okay, then you have a big problem. All right, so please search for questions, gain more confidence. All right, so now this is a 2023 question. Number 18. It says that in monohybrid inheritance, if an organism carries two different alleles for a particular gene, it is called two different alleles for a particular gene. Okay, two different, dissimilar what? Alleles. What's it called? Is it heterozygous, homozygous, dominant, or recessive? You know the answer now. Tell me the answer in the comment section. Then 2022, number 23. It said dash is the inheritance of characteristics controlled by a single gene. Inheritance of what? Of characteristics that is controlled by a single gene. He said dihybrid inheritance, B say monohybrid, C say single inheritance, D say double inheritance. Inheritance of characteristics controlled by a single gene. We're looking at what? One character, okay? One character, all right? Is it mono or di? Tell me the answer in the comment section. So 2020 now, number 27. It says, when gametes from pure breeding plants with contrasting features such as tallness and shortness are involved in monohybrid cross, the offspring in the first filial generation are usually what? A say pure breed, B say heterozygous dominant, C say homozygous recessive, D says mutant. Okay, so the offspring in the first filial generation, we don't know what? They were tall now, Abi? Okay, they were tall. Were they heterozygous tall or homozygous tall? Tell me the answer in the comment section. 2007, number 47. It says that use the diagram below to answer the question. You are seeing what the diagram, all right? You are given what 4 dogs. They say if the dogs are offspring of a monohybrid cross, all right, and the gene G for gray head is dominant over its allele GA, okay, the individual whose genotype is likely to be GG, okay, is what? If you look at the diagram from the phenotype, okay, you can see already, okay, the one that have what? Gray head, all right? So now, from our first filial generation and second filial generation, okay, we had what? A pure tall plant, okay, for example, okay, so if you are using what? You are using the gray head now for the dog and GG. So we had what? GG, we have GG, we have GG, then we have what? GG, okay? All right? So that is what our result of what? Of the cross, all right? That will be our result. Now they're telling us that the individual whose genotype is likely to be GG is what? Which is what? Pure breed, okay? That is not gray head. From the diagram, all of them have what? Patches of what? Of gray around them, apart from what? From the fourth one, okay? That means obviously that will be our answer there. From the skin color of the dog, you can see, all right? So just tell me the answer in the comment section. I've explained a lot. Now, 2021, number 40. They said the genotypic ratio, 1:2:1 in the offspring of a hybrid cross illustrates the law of what? A say use and disuse, B say dominance, C segregation, D linkage. Okay? Tell me the answer in the comment section. 1:2:1. At the end of monohybrid cross, that is what we had. Now, you search for 2020, number 11. Tell me the answer in the comment section. 2018, number 26. Tell me the answer in the comment section. 2006, number 23. Tell me the answer in the comment section. Okay. All right. There are so many other questions from the app. Download the app. Okay. Help yourself succeed at once. Okay. I wish you all the best. My name is Master T. I'm the Grand Commander of the Old Risco Army. Thank you for joining us. We are very glad you joined us, and we are very eagerly waiting your testimonies. See you in the next class.